Mammalian tidal lungs evolved as outpocketings from the back of the pharynx. They evolved as little pockets that hold oxygen with capillaries nearby to pick up that oxygen. But a problem is that they're not very efficient at extracting that oxygen. And that's what we'll go through when we talk about mammalian and other types of vertebrate tidal lungs. In a mammal, the structures are complex, going from the air coming in through the mouth, through the larynx, through the trachea. They're stiffened with cartilage, so they form a rigid structure that is hard to collapse. The air moves into the lung. We call them tidal lungs because when the lung expands, as the chest cavity expands, that moves air out. When the lung compresses, it simply goes back along the same paths, like the tides moving out and then the tides moving back in. So air comes in and then goes out. In the cartilaginous area, there's epithelial tissue that is much thicker than you're going to see when we look at the lung tissue itself, lined with cells that include mucus glands that are secreting mucus that help trap particles in the airway, and stiffening structures, the cartilage here, connective tissue. If we looked at one little patch of the lung here, each branch goes as a bronchiole down to what look like little clusters of grapes on a branch. These little clusters are alveoli. Each alveolus is near capillaries, that is lined with capillaries, with deoxygenated blood coming from the heart, from the pulmonary artery, picking up oxygen and returning to the heart to be sent to the system. The result is a very large surface area. The lung of a human has the surface area approximately that of a tennis court. In contrast, the lung of some other animals, frogs for example, have a lung that is a simple sac with ridges, a little more than a bag with blood vessels all along the surface. Zooming in here, here's the individual alveoli again with blood vessels around them. If we just took one and blew it up and made a cut through it, what we'd see are the individual epithelial cells here with nuclei. There would be alveolar pores, the opening going in. Here's this large surface area of cells. Notice how thin each cell is when we cut through it. And some macrophages in there. These are amoeboid cells that are wandering around looking for anything from dust particles to bacteria invading organisms. In addition, there'd be cells that are having other secretory functions, including coatings to keep the surface moist. So the respiratory membrane is here. Capillaries run along the surface. This would be a single red blood cell inside this capillary. This is a slice taken through the alveoli. Here you see clusters of alveoli with individual alveoli. Here's an individual alveolus, here's an individual, here's the opening into that alveolus. Here's the bronchiole, which is branching into all these little clusters of grapes. One of the things I hope you see is how thin these areas are. We have areas of larger blood vessels, and the very smallest of these are capillaries. Even if I blow them up, you can barely see them. These thickened areas here are capillaries. These larger blood vessels, each alveolus will have capillaries dotted around it. Here's another way to represent a tidal lung with a breath and milliliters of air inside the lung. For a large human, the total might be 6 liters of air. A typical breath, when you're just sitting or as I'm sitting here talking, a typical breath is moving only a very small amount of air in and out. And that's called the tidal volume. It's the amount you normally breathe when you're not working hard. In other words, you're using only a tiny fraction. In this case, it's less than a tenth of the full capacity of the lung. A typical mammal can't afford to collapse the lung completely because the fluid, the liquid, would tend to glue it shut. Hydrogen bonding of the water on the surface of your lungs and of the surfactants would have a tendency to stick, making it very hard to inflate the lung. So when you exhale, you keep the trachea, the bronchioles, all of that remains open and the alveoli don't completely collapse. This residual volume has O2 depleted air. If you take a very deep breath, inhale to your fullest, you use all of your inspiratory reserve. In other words, if I'm sitting here and I take a full deep breath, that brings me to the edge of my inspiratory reserve. If I exhale all that I can past what I normally would, that takes me all the way down to the edge of my expiratory reserve. What I cannot exhale, bronchioles, bronchi, trachea, that's the residual volume. With normal breathing, you're doing this. When you're working hard, so running hard or exercising hard, you're taking deep breaths, exhaling it all, taking a new deep breath, exhaling it all. Even under those conditions, a good 10 to 20% of your lungs remains with air inside it that is O2 depleted and carbon dioxide rich. So what I've drawn here is a human with air coming into the mouth, air in the lungs, blood coming in that is deoxygenated and leaving oxygenated. Now the air coming in is at 160 millimeters of mercury partial pressure of oxygen, or 20 kilopascals. As it comes in, it begins to mix with the oxygen-depleted air that's already there in your residual. And so by the time the air gets into your lungs, a typical amount of oxygen might be 105 millimeters of mercury partial pressure oxygen. If you breathe deeply and fast, it'll be higher. If you're breathing more slowly, it can be lower. We're only at 2/3 of atmospheric partial pressures of oxygen. Some of that oxygen is being picked up and carried away in this oxygenated blood. In a tidal lung, as the oxygen is moving out, total partial pressure of oxygen inside the lung is getting lower. At some point, you exhale that air and bring in fresh air, and we start over at 105. The partial pressure of oxygen that your blood is encountering is in the range of 105 or so. So to summarize, a mammalian lung exhales depleted air that is somewhere below 80, perhaps 50 or 60 millimeters of mercury, and then inhales air that mixes going in with the partially depleted air, 105 millimeters of mercury partial pressure of oxygen. The system, deoxygenated blood coming in, can pick up oxygen that is at most 105. As it gets depleted, it goes down toward 80 and below. And so the blood leaving is somewhere in the range of a partial pressure of 80 to 105 millimeters of mercury of oxygen. It's about 100% saturated with oxygen. An important point here, though, is that your blood will never be at 160 millimeters mercury of oxygen. Unlike fish that can get almost all the oxygen out, you continually are exhaling air that still has significant oxygen in it. You can hold your breath and get more oxygen out of it, but that actually means you'd pick up less than if you could keep breathing.